I'm Shunichiro Tomura, who is doing a PhD with Mark and Owen. And today's presentation is about the ensemble approach for gene prediction in crop breeding. So I'm going to introduce brief background information at the beginning so that we all can understand my research target is. And after that, I'm going to introduce one method to tackle this my research target, which will be discussed in evaluated and experimental results section. And after that, I'm going to briefly summarize key findings from this talk. So let's move to the my first section of the presentation. So as we all know, the food security is one of the significant issues in this world, which is caused by two different components. The first one is the increased population. It's said that by the end of 2050, the population will be more than 9 billion people in this world, which can lead to the increase in this food demand. And another component is extreme weather events caused by the climate change, such as drought, flood, heat wave, and then extreme like severe thunderstorm and so on. So those extreme weather events can lead to the devastating damage to the crop yield, and then this can lead to the decrease in the food supply. That's why we think about the one scenario where we have to increase more food production under the extreme weather events like this. So this is a very difficult issue and then difficult to solve this task. And then there are many ways to tackle these problems. And then one approach can be the application of crop breeding by developing new cultivars which can produce more crop yield under the harsher and the more difficult environments. So next, let me briefly explain about the breeding cycle so that we can understand the significance of the breeding programs. So suppose we have one population which has a number of different individuals. And then after that, we try to measure the phenotypes of the different individuals. And if you see distribution, it can be something like this. So this vertical line, the distribution shows the mean value of the phenotype. And this sigma shows the variation of the phenotypes caused by the genetic marker differences here. And from this population, we tried to select the top K individuals having the most desirable phenotypes. And this selection level is called the selection intensity, represents this small I here. And after the selection of the different individual plants, we have to grow them in the different environments. So this is called the multi-environment trial. And the selection of the environment is based on target population of environments, which are the environments that are actually going to be used for the commercial use in the future. And then after the phenotyping and then the data collection from those different trials, we tried to, again, select the top individuals having the most desirable phenotypes here. This selection accuracy is represented as this small r. And after the selection, we cross them so that we can produce progeny. And if you measure the phenotypes again, like before, the distribution can be something like this by moving the distribution from the left to right. So this gap from the previous and the current distribution is called the gene gain. So this ultimate goal for breeders is to increase this gene gain as much as possible. So this is the significance of the breeders and the breeding companies. And this is one typical example of the breeding cycle. And then the length of the cycle is represented as this capital L here. So to sum up, so this gene gain can be influenced by the selection intensity, selection accuracy, and genetic variation and breeding cycle time. And my question here is that this gene gain, it may not be large enough to solve the food production and food security issue, given the food demand increase and food supply decrease, like I mentioned before. That's why we have to think about some other ways to accelerate this gene gain increase. And then one approach can be the application of gene selection. So one major difference from the previous traditional breeding program is the application of this process called gene prediction, which replaces the manual phenotyping phase with the prediction process. So since this gene prediction is very important to understand gene selection, so let me explain about this a bit more by using this simple diagram here. So again, suppose we have one population, and which has a number of different individuals, and then from a part of them, we try to extract the genetic marker information by DNA sequencing. And then if we focus on the extracted information, the majority of the part shows the similar information, but some part shows different allele combinations. So those parts are called the single nucleotide polymorphism. Then and for my research, I just use this SNP effect for the genetic marker information here. And also, we extract the phenotype information from the selected individuals from this, and so that we can convert the information into the tabular format called labeled data, which has a SNP information and their corresponding phenotypes. And by using this data, we can try to train the prediction model. And after the training part, we extract the DNA information from the remaining of the population, so that we can extract the genotype information and the DNA information to have a SNP importance, SNP information here, which can lead to the development of the unlabeled data like this. And then by using this unlabeled data, we input this data to the prediction model to target prediction of the target individuals here. This prediction value is called the genetic estimated breeding values, GBVs. And then for the internal target trait, we have different targets like tail number, flying time, height, and so on. So this is a very simple flow of the gene prediction. And then by using this gene selection and the prediction approach, we can provide a positive effect towards the breeder's equation by reducing the breeding cycle time and increasing the selection intensity by scaling up the population size for the breeding programs. And this can lead to the increase in this genetic gain, which is one of the significant targets for the center to increase the food production in the future. However, we can further expect to increase this genetic gain by increasing the selection accuracy by developing a new novel approaches and prediction models to increase its performance, which is going to be my research target. So for my PhD, I'm going to focus on how to increase this selection accuracy by developing a new prediction approaches. So this is my research target for my PhD. Let's move to the next section, method part. So in gene prediction for crop breeding, we have a bunch of different prediction algorithms. So one typical example can be this. So for example, we can develop like a genetic marker information matrix to predict the target phenotypes by using a genome-based linear unbiased prediction GBAP method. Well, maybe some people may want to even further this approach by using the idea of the prior and posterior distribution to further increase the performance through the Bayesian approach. And or maybe some people even further try to map that point into the higher dimensions to capture the non-linear interactions between that points by using the reproducing chronology by space or KHS. So those methods are called the common statistical methods because they have been used for a long time since the beginning of gene prediction. However, we also have different approaches like this. So this supported vector regression draws the hyperplane between data points to predict the target phenotypes. Or maybe some people want to apply this neural network approach to capture more complicated interactions between data points by mimicking human brain structure. Or maybe some people want to develop a bunch of different like decision mechanisms to further enhance the prediction performance via random forest approach. So those methods are called machine learning models. And for my PhD, I like to focus on those two different categories of prediction models here. And then in many cases, we try to find the best single model from them, which can outperform the others all the time, regardless of the scenarios and then trait and so on. However, this approach hasn't been that successful because there's no constant winner caused by the diversity in prediction and the information capturing process. So, for example, maybe for one case, maybe this GBAP approach can be able to achieve the highest performance across the different traits for one data set, but it may not be true for other data set and so on. And also, there's no complete prediction models which can capture all the perfect SNP effect estimation. That's why, as a result, it's kind of difficult to know which model should be used for the improvement of the prediction performance. That's why we have to think about a new way to increase the operation performance in a stable way by somehow using all the different information captured by different single models. And what should we do? And then one approach can be this method called ensemble approach. So in this case, instead of competing in each other to be the best single model, perhaps maybe they can collaborate each other by giving the piece of information, and then at the end we can aggregate all the different information into one to further enhance the operation performance here. So this is briefly explain the approach for the ensemble in a conceptual way, but now we have to evaluate and then test this approach by actually implementing it by using a real data set, so which is going to be in the next section, explain the results. And then in this talk, I choose one data set called TNM data set collected by Chen et al. In 2019. This is a publicly available high-quality data developed by the hybrid of the maize and teosinte. And this includes both genetic marker and phenotype information, so that we can do gene prediction for our experiment here. And for in terms of target trait, I chose two different traits, D20 and serial number here. I chose those traits because those are significantly important for the crop yield prediction, and then therefore the center has targeted the investigation of those traits as an important trait for the research. That's why I chose those two traits to investigate. And then let's move to the next part, which is the actual implementation method as a one example in this case. So suppose we have one data set containing training test data set here. And then by using this training part, we try to train each of the single models like this. In this case, we have 6 different single models. And then after the training, we have to evaluate their performance by using the test data set here. And then during this process, we can also extract the prediction result for the test data set, and which will be assembled into one to develop a prediction value matrix. This matrix is going to be used for the input for the ensemble algorithm. And then in terms of the actual implementation path for the ensemble approach, we leveraged one approach called the naïvely averaging method, which simply take all the prediction result from all the single models, and then we just simply average them into one by using the same weight. And similarly, we also have to evaluate this performance by using test data set. And then in terms of evaluation, we use one method called a metric called the Pearson error correlation. So in this case, a higher value in the Pearson correlation represent a higher prediction result. Prediction result, which can directly lead to the increase in the selection accuracy in the breeder's equation. And then let's move to the actual result section part here. So this is the prediction result for the both trait, D20 and serial number, when we use the single models individually. And each box plot has 6 different boxes representing their performance from left to right. These are the GPAP method, Bayesian model, RKHS, random forest scaffold vector aggregation, and then neural network here. And as we can see that in many cases, single models perform similarly. That's why it's kind of difficult for us to pick which model we should use to predict the target phenotypes here. That's why it's kind of difficult to further enhance the selection accuracy and genetic gain as a result. And now here comes the ensemble approach here, colored by red here, red boxes like this. It's obvious that the position of this ensemble approach is higher than any of the prediction models, indicating that this ensemble approach was able to outperform others in both traits here, which lead to the increase in selection accuracy and then genetic gain also as well as a consequence of this. One interesting finding here is that although we simply averages all the prediction result, its performance is not averaged by having the higher position here, as we see like this. So this is an interesting finding for the from the ensemble approach here. And then from those findings, we can have one big question, which is why was ensemble approach able to improve this performance? So there are many ways to think about this, and then one hypothesis can be the aggregation of the distinct information for predictions. And like I said in the previous slide, Each of the single models captures different sleep effect in a different way by using their own mechanisms and algorithms. Therefore, by aggregating all the captured information into one, by combining all the information at the end, and we can complement the information each other to have a more like a complete information for prediction, leading to the further improvement of the prediction performance in the end. So this is the understanding, hypothetical explanation for the ensemble approach, but now we have to test whether this explanation is true or not by using the result from the order set. And to do this, generated pairwise comparison between the single prediction models here by developing a scatter matrix here. So this matrix has two different main parts. The first one is from the top triangle part with the green color representing prediction value. This one is representing the pairwise comparison between the n of the single models at the level of the prediction values. So P represents the phenotype level, whereas this bottom triangle part colored in the blue and orange goes one step further by doing a pairwise comparison between the single models at the level of sniff effect. So this bottom part goes one step further to find out the mechanisms of the predictions from each of the single models here. And from this, we can find two main key findings. The first finding is from this top triangle part, showing that a linear trend at the 45-degree angle in many cases like this. It's representing that each of the single models return the similar prediction result, although the same input was given to all the different single models here. This kind of supports the idea of why we get the similar prediction result for the result and performance for the prediction performance in the previous few slides. So this is simply because they return the similar prediction values in the end, but although they are very different from each other. So this is the first key findings from the top part. The second key findings from the bottom part, showing that the little or no trend here. This is kind of interesting finding, because although each of the prediction models return the similar prediction performance with the same like input data, their prediction mechanisms are quite distinct from each other by estimating a sniff effect in a different way. So this kind of supports the idea of why we get the performance improvement by using the ensemble approach. This is because the ensemble approach can combine all the different sniff effects into one by complementing them into each other at the end. And then that's why we can enhance the prediction performance at the end. This supports the hypothesis that I mentioned in the previous slide. So those two key findings can be supported by two interesting concepts. First one is the null fluent theorem. It's saying that the performance of the single models become the same on average. That's why all the different single models was able to return the same prediction values in the end. And then also we can say that it's kind of difficult to pick the best single models, because this theorem is saying that there's no such a model. It can be the best of the time. And then from the bottom part, this result is supported by the one concept called the Rashomon effect, saying that there's a set of models that have the similar prediction performance, but their calculation mechanism is different from the same data. So by those two key findings, we can find the strength of the ensemble approach. So from those interesting key findings, we can think about, we can point out three different novel points here from our research. The first is the application of the ensemble approach in gene prediction for crop breeding. The second one is the use of both non-QTL and QTL for genomic prediction. So I was able to achieve the relatively higher prediction accuracy for the both traits by using both single and then ensemble approaches. But without the use of the non-QTL, I wouldn't be able to achieve such a high prediction performance. That's why it's better to use both more comprehensive like sigma marker information to further achieve the prediction performance enhancement for gene prediction. And the last one is the most significant part, which is about the investigation of the power of the ensemble approach. So like I mentioned in the previous slide, when the non-fluence theorem and the Gresham effect are effective in the prediction result, then there's a chance that we can further enhance the prediction performance by using the ensemble approach. So those are the novel points for our research at this point. So in summary, we can say that there's potential that ensemble approach can improve the prediction performance on the gene prediction for crop breeding, which is attributed to the aggregation and the combination of the distinct information from each of the single models. And for the future work, we have main 4 points at this moment. The first one is the application of the ensemble approach to different data sets so that we can check whether we can generalize our findings to other data sets. The second point is the application of the ensemble directly using sieve effect. And at this moment, I just simply averaged all the prediction result by using all the phenotype prediction result. But we can also think about directly averaging the sieve effect so that we can use that averaged information to further improve the prediction performance by revolting the prediction models here. And thirdly, maybe we can further improve the performance by optimizing weight when we do averaging. And I believe that one model can be more informative than the others. Therefore, by identifying those are more significant single models and then put the correct weight on each of them, we can further try to enhance the prediction performance. And lastly, we can also further investigate the gene variation parts here. This, like I showed in the scatter plot matrix, each of the single models return the different sieve effect estimation. Therefore, by using all the different information, and we may be able to further investigate the way to further enhance the gene variation in this breed association represented as a sigma here, so that we can also try to enhance the gene gain in the end. And so that's the end of my presentation. Thank you for listening.